Fellows and members of IVA and all of those who are interested in favorite subjects like diesel fuel and cancer medicine are most welcome to this IVA Science and Society Forum. My name is Leif Johansson and I'm chairman of the IVA organization. And as you remember, the longer name of IVA is the Royal Academy of Swedish Engineering and Economic Sciences. So it has a broad portfolio of things that we want to cover with our different activities. Now, tonight we are starting with IVA Science and Society Forum, which was created to allow the very good scientists, and indeed we have one here tonight that Björn, our president, is going to introduce in a short while, to allow them to really explore what the frontier of science and technology can actually do to society and what will be the impact on society. And we, obviously, as an academy who are much involved in the interface between society and science, that's one of our areas of real interest, and the ability for scientists to talk about in more popular terms what they're doing, we think is good. It's certainly good for society. I would expect that it's sometimes good for scientists also. But it's an important aspect of how to fund and actually generate resources for science. Now, as you might know, I've been the CEO of the Volvo Group for 15 years, and I've now been chairman of AstraZeneca for 7 years, I think it is. And therefore, when someone invites me to a seminar that says we now have someone who can talk to us how we can make future diesel fuels, then that covers my Volvo experience very well. And on top of that, throws in how we can do better biomarkers for cancer. Then I think you have my absolute interest for tonight's seminar. So with that, let me wish all of you welcome and hand over to Björn, our president.
Thank you, Leif. It's my pleasure to give some opening comments for this Science and Society seminar. And my name is Björn Nielsen. I'm president of this academy all until Wednesday. It's my pleasure. Today we're going to hear Jens Nielsen talk about what do biofuels and cancer have in common. And I will actually start the seminar by giving some introduction by presenting some words about IVA, what we're doing, what our portfolio looks like. So that is in just some words. Currently, innovation is a buzzword. We need innovation in our society, and if you take the burden to Google it, which maybe you shouldn't, you get 528 million hits, which means you get it everywhere. I haven't looked on all of these hits. If you take mathematics, you get around 300 million. So it's more commonly used. Of course, it can be used in many different contexts, but many of these are exactly in the kind of context I mean here. The reason is, of course, that innovation, to find solution to master of grand societal challenges, is tremendous. We put a lot of hope in knowledge, that knowledge should come to use and resolve many of the big issues we have. And this is nothing new in a sense, but it has been accelerated. We also, of course, attach it to financial growth, to prosperity, for jobs, and I think it's a good hope. Maybe it's the only hope we have, but it's not a bad one. And already 1971, Alan Kay put the following very nice words: the best way to predict the future is to invent it. And I think that is a very strong language and something that I definitely subscribe to, and I think it's related to this topic. And it's also highly linked to engineering. Theodor von Karman was an Hungarian. He was an aeronautics mathematician. He made the mathematics behind these turbulence around wings, so that was the foundation for jet planes, and later to really model that. He was active at Caltech, but he was an Hungarian. He put the following phrase, because he wanted to understand how knowledge is built and how what is then engineering? And today we're going to hear about engineering. And he said the following, which I think is poetic. Science studies what is. Engineering creates what never was. Note the last twist there. Engineering creates what never was. And that is the connection, and also that our sister academy in the US, National Academy of Engineering, really use as a tagline to understand, because many scientists are also engineers, because they do create maybe their own methodology in order to study nature. So engineers, we create something new. Here, IVA is perfectly positioned. I would say IVA was created in 1919, but has never been more modern than today. Founded as the first engineering science academy in the world. Axel F. Enström is our founder. And the way we should operate is to promote the engineering and economical sciences, as Leif alerted to, and the advancement of business and industry for the benefit of society. So it couldn't be more modern. It's the science, it's the business, and the benefit of society. The way we do this, we are structured with our fellows. We have national 12 divisions. We have also international fellows, around 250. We have also a business executive council that time does not allow me to go into, but we have companies, around 250 member organizations, mainly corporate organizations, are associated to the academy, consisting of the fellows. We also have an executive committee that I will show here, which is Leif is chairing, and we have 8 members of. We have the 12 divisions, where many of the chairpersons are here today, and this is the foundation. This is an academy. When we want to have an impact in society, which we do, then we start projects. Then the project have some feature. They're based on scientific. It's a knowledge approach. The goal is to influence society. We start projects in area where we want to influence politics. Often controversial topics like energy, what kind of electricity supply can we have in Sweden in the future? Very controversial, but in the academy not controversial at all. Often vested interests, because then an academy as an independent body can contribute. And we involve, of course, the strong network of scientists, business leaders, and decision makers. So currently we are running a large number of projects. The pre-projects are the projects to be. The projects are the ones that we're running now. The ones in yellow are the ones currently started very recently, and the yellow to the left are the ones we expect to be started at least before the end of the year. The long-term activities I want to highlight, Work Leap and Technology Leap. Both they are sizable, and there are projects ongoing where we, for the Technology Leap, we interest young people for science and technology, and the Work Leap is for integration of highly educated immigrants, often refugees. I also like to mention we do a lot of things in addition to these projects. We, of course, have within the academy the basic organization for seminars. We also conduct Royal Technology missions. We were in the US in May. Next year the plan is to go to Hong Kong and mainland China in the Pearl River Delta. So in about a year from now. We also do a lot of things. We publish numerous reports, and we also have more than 100 seminars per year. We had in January Angela Merkel elected to come here as her only public appearance when she made a visit to our government. And that was a big happening that Johan Weigelt organized together with the Swedish-German Trade Council. So with that, that was enough of IVA today. I can talk forever about IVA, but I do not intend to do that, because we will have a seminar. We will have Jens Nielsen to present what do biofuels and cancer have in common. Professor Nielsen will be, Nilsson sounds almost better. Jens Nielsen will become an IVA gold medalist tomorrow. He's been a fellow of this academy since 2010. He came, I think, 2008 to Chalmers from DTU in Denmark. And you had an associate professorship with the KTH. I think it just expired, but you're definitely scientifically very much linked to KTH research. And you're unique and well-decorated scientist. And what I like about Jens' research is that it's really based on fundamental research. You really are there to dig, to understand what you're doing, and then you apply it and do something with it. And I think that's why you have received many honors. Tomorrow will, of course, be the greatest with the IVA gold medalist. So I'm really looking forward to your talk. So please give a warm hand to Jens Nielsen.
Yeah, thank you, Björn. Thank you very much for the kind introduction, and I'm of course also deeply honored for the gold medal that I will receive tomorrow. I would agree that is probably one of the biggest honors, or maybe the biggest honor that I have received, and that will be a fantastic day. I'm also very, very happy about the invitation to give a talk here today, and I will try to find a title that could have a broad interest. It was not just to inspire the chairman of IVA to come, but I would also give a little bit of background about the field of biotechnology. Maybe I can already start to reveal what biofuels and cancer have in common, and that is metabolism. Metabolism is the core of all cellular function because it provides the building block for the cell. So this is like the feedstock for building anything. It also provides the free energy, so it's like an energy engine that fuels the cells. And then it also needs to handle electron flows. And one could actually put some numbers behind some of these flows that are actually happening in our individual cells in the body, and they're kind of staggering. And we can use knowledge about metabolism and its regulation to engineer cell factories, and that's what I will be talking about in the first part, which will be the longest part of the lecture. But we can also use this to diagnose diseases and predict its development, and that's where we're going to focus particularly on cancer. And at the end, I will also show some work that we have been doing in order to look into if we can improve the energy uptake from food. That is not something that we here in the Western world are particularly interested in. We are actually having too good uptake of energy from our food, but there is a major problem, particularly in Africa, but also many parts of Asia, where a lot of children are stunted because they don't have enough energy uptake from their food, and that has significant impact on their cognitive development going forward. So let's look a little bit on metabolism as a very brief introduction. And what I've drawn here is a yeast cell schematically that converts glucose as the main sugar that yeast is growing on, and converts that into ethanol, and it does that very efficiently, and we rely on that in making bread, beer, and wine. In bread, of course, the ethanol evaporates as soon as we put the dough into the oven, but associated with the ethanol, there's a very large and rapid CO2 production, and that made the bread raise. But of course, we're also relying on this for making bioethanol that is used as a substitute in gasoline. But you can see that the scaffold of metabolism here is very similar to what we observe in a muscle cell here. It's also taking up glucose and it's relying on this. But if it's lacking oxygen, it will produce lactic acid. So it's a different product, but it's a very similar scaffold. And so just this simple illustration points to this similarity that we can use. Actually, this overflow to lactic acid is also happening in cancer cell, and there it's often referred to as a very classical Warburg effect. So if we look a little bit back on biotechnology and studies of metabolism, it's really ancient use of cells for production of beer and wine. There are reports going back more than 10,000 years where yeast were used for raising breads. And this has been illustrated in a number of drawings. But most believe that it's probably around this time here, about 5,000 years ago, that this started to be developed significantly. At that time, of course, there was no knowledge about what was making the ethanol, but that was shown in some very critical experiment by Pasteur in the 18th century, 19th century, where he showed that if he takes a broth that has a lot of nutrients in it, and he sterilizes and closes the flask and sterilizes this broth by heating, then he can see that there's no conversion of the nutrients in this broth here if the flask is closed. But if he breaks it open, microorganisms will enter this open flask and result in fermentation. So this very simple experiment here really led to the identification that this is not a spontaneous chemical conversion process that is happening, but it's actually living cells that are involved in this conversion. This led a Danish researcher, Emil Christian Hansen, who was working at the Carlsberg laboratory, which was established by the founder of Carlsberg, to do actually basic research in chemistry and physiology. And Emil Christian Hansen was the first to isolate yeast and show that this was responsible for beer fermentation. And that allowed to actually significantly advance beer production, because suddenly you did not have a lot of spoilage processes, and you could start to use an inoculation of a pure yeast culture for making the beer. And that is what is used today in both beer and wine production. A few years later, James Weissman was the first to, probably the first to demonstrate the use of microorganisms for production of chemicals. So he developed the so-called acetone butanol ethanol fermentation process, and that was interesting because particularly during World War I there was a large need for acetone in the production of explosives, and you could then rely on this microbial fermentation process. He identified that this process was carried out by a bacterium called Clostridium acetobotulicum, and that he could then patent, and that resulted in establishment of a process in both in the US and in the UK. Interesting enough, this is not the first time when science and politics went hand in hand, because he actually met the UK Prime Minister Balfour and donated the rights to use this process in UK when the return that Balfour made, the Balfour Declaration, which eventually resulted in the establishment of the State of Israel. Another key landmark in what we could call industrial biotechnology was the discovery of penicillin. That was discovered by Alexander Fleming in 1929. Another beautiful example, you can say, of a scientist that had a discovery and instead of just discarding it, actually pursued and followed on this. Because what Alexander Fleming was doing, he was actually looking into lysozyme, an enzyme that degrades bacteria. But one day, following a vacation, he came in and he observed a contamination on his plate. And here he has the bacteria growing that he was interested to cultivate for his studies. But he had a contamination of a fungus that had grown on the same plate. And what he was observing was that around that fungus, there was very little growth of the bacteria. And so many people would probably have seen this as a failed experiment, thrown out that plate, but Alexander Fleming actually pursued this and speculated that this fungus may actually produce a chemical that is killing these bacteria, and that led him to discover penicillin. And over time, that is probably the most life-saving drugs of all times, penicillin. And even today, we tend to underestimate the value of antibiotics, particularly in the Western world, but really also in the developing world. Antibiotics is a really life-saving drug in many, many cases. Following discovery of penicillin, that led to what we often refer to as the golden age of antibiotics. Selman Waxman identified streptomyces in 1943, and that could be used for treatment of tuberculosis. This was not a fungus, but a bacterium that is producing this. Actually today, this class of bacteria, streptomyces, is used for production of a whole range of antibiotics and also other bioactives. And so this led to that in the 50s, in the 1950s, there was really all major pharmaceutical companies were starting to produce antibiotics. This went on until about in the mid 80s or something like that, when actually most antibiotic production started to be outsourced to India and China. And today, practically all antibiotics is produced in China and India. It's a little bit interesting to look into the current use of antibiotics, because I mentioned this extreme value of antibiotics, a drug that's a class of drugs that we take somewhat for granted, and this plot here illustrates quite well that antibiotics is often the first thing that comes into healthcare sector when it advances. And this is seen here in a plot on the standard units or intake of antibiotics per GDP per capita here. We can see that it's relatively constant. So even when you have a relatively low GDP, you actually reach Western standards in antibiotics intake. There are a few outliers here, France, UK, and US, that have an overuse of antibiotics, causing also major problems with antibiotic resistance in hospitals. So, but this is really an interesting example. But particularly the overuse of antibiotics, but also in general, there is a constant need for new antibiotics, and I'll come back to that briefly also later on in my talk. Later on in the biotech development, it became possible to clone genes. That means to take a gene from one organism and put it into another organism and use that organism then to produce a protein. And one of the first examples of that was to produce human growth hormone, which before that was extracted from calf organs, as well as also human insulin, which was extracted from pig pancreas. And of course, you can imagine also that this was a much cleaner technology, as well as it actually allowed to produce the actual human proteins that have lesser side effects than the traditional extracted proteins. And today, the biotech industry, as we often refer it to, is a very large industry, more than $200 billion in annual sales, and falling in many different categories. And particularly in recent years, this has experienced a very strong development in the development of particularly antibodies for cancer treatment. So the current use of cell factories is really broad. We have it in classical products, food and beverages, in pharmaceuticals, a whole range of different types of pharmaceuticals, and fuels and chemicals. In this group here, as I mentioned, this is really with a very rapid growth here. But also, if you look at the production of chemicals, it is still very small. It's only about 1% of the total chemical production volume out of an industry of about $3,000 billion US. It's only about 32 billion that is renewable chemicals. But this is rising much faster than the traditional chemical synthesis, and so it's expected actually already by 2020 that this will pass 80 million. So how is this done? Well, particularly for renewable chemicals, what we're doing is that we are relying on recruitment of a cell factory. So we can have yeast, for example. Here is a microscopic picture of yeast that takes the raw material, and the raw material, the feedstock we're using in all these cases is sugars. It can be glucose, sucrose, or xylose, depending on the source of that. And we're converting that into the product of interest. And you can imagine that we could recruit a cell factory that naturally produce a product we're interested in, like yeast that is producing ethanol. But we can also take yeast and engineer it and remove its capabilities of producing ethanol and inserting new enzymes into this and hereby produce the new product. And this is what we call genome engineering. We change the metabolism of the cell such that we can now recruit that cell to produce the product that we are interested in. So the interesting thing about this is that this is consistent with this so-called biorefinery concept, and we can have a very equal analogy with how the chemical industry actually evolved over time. They were relying on oil. We are relying on plant biomass as feedstock here. The chemical industry have taken oil and cracked it and used catalysis to convert different components from the oil into different products out in the end. We are relying on first cracking of the polymers in the biomass into sugars, but then our catalysis is a living cell that performs this conversion and into the different products that we are interested over here. So it's the analogy is very similar to a traditional refinery here, but we're relying on this catalyst here. So let's look a little bit on the challenge, because this sounds relatively easy. So let's look at actually the penicillin development process, because in order to develop a cost-competitive production process, it's really necessary to improve the performance of the cell factory. And here is shown the performance of Fleming's original strain. And please note that this here is an exponential axis here. So this was really very, very low production, that his strain. And actually, in order to get enough penicillin for the first clinical trials, they were extracting penicillin from the urine of the patients and reusing it because they simply couldn't get enough of it. But they managed to relatively easily isolate a new strain that was better, that was mutatinized, and actually that resulted in a rapid improvement here. And then over the years, this penicillin production has been improved, and basically you can say you have improved the performance of the catalyst over these years here. And this looks pretty impressive here, but also notice that this is a lot of years that has happened here. This is something that's been going on for 60 years. And we cannot really wait 60 years if you're going to do something about climate change. We really have to speed up this process here. So why does it take so long time to improve the performance of the catalyst? And why is it so complicated to improve production? This is the reason. Because metabolism inside the cells, it sounds easy to recruit it, but it involves a very, very large number of reactions. Actually, in a yeast cell, there's about 2,000 of these. They are associated with individual genes. Each of these genes are encoding an enzyme that is a catalyst for each of these reactions. So we have this very large metabolic network and set of reactions that have to operate in a coordinated fashion in taking the sugar and convert it into the product of interest. If we look into a human cell, we are more complicated, and in that sense, we have many more reactions. Many of these reactions are particularly also because we have to deal with, for example, a number of different complex products that we are eating, like food, and degrading, and also detoxify, for example, different toxins and so on. So this is the classical view of metabolism here. That we see here, and for those of us who have studied biochemistry, have looked into individual pathways and learned this in this way. But it turns out that metabolism is even more complex than just the large number of reactions, because it's highly connected. So you can imagine that it's really like a metro network that we are suddenly going to redirect the activity of some of these lines in a different direction, and that will of course have impact on the whole traffic scheduling in that line. So this is just an illustration of where many of these reactions are. The green dots here are the enzymes, and all the colored dots are metabolites that are interconverted in these different reactions. And that's happening in different compartments. And it's kind of obvious that if we are, and I said it kind of easily, that we delete ethanol production, we may delete a couple of enzymes here involved in ethanol production, but you can see that may perturb and change the activity of many other reactions inside the cell because it's so highly connected. So how can we study such a complex system? Well, here we are helped by the genomics revolution, because that has really changed things. Actually, already back in the late 80s and early 90s, there was much interest in doing these kind of things. ICI had a big initiative. Many different chemical companies have initiatives in order to produce chemicals using microbial fermentation. But most of these initiatives failed because it was simply the technology was not good enough at that time. But today, particularly because of genomics and a number of different tools that really allows us to study metabolism in a much deeper sense, we often refer to this as systems biology, we can begin to get a grasp on this and thereby also change it. So you can imagine it's a little bit like this. In the early days, we had Pasteur knew that substrate was coming in, sugar was coming in, ethanol was coming out of the cell, but had no idea what was going on. It was a black box, well, actually here in this case a white box. But today we can add information about what sequence is there in the genome of this organism, which genes are actually expressed at different conditions, which proteins, and these proteins are the catalyst for specific reactions, and we can also understand the individual reactions and so on. So this is really technologies that has been developed over the last 10, 15 years that allows us to query this complex system in much larger detail than we could ever do before. So coming back to now how we can recruit this technology in this design. And here we are approaching this just like in any other engineering discipline. So we're laying out a design principle, we are building this, but as our design and our knowledge of the biology is not 100% yet, we have a lot of enzymes inside the cell that we do not know what they are doing. Therefore, we have to test our build. I guess Volvo Cars is also testing their designs when they have built them, but that test is normally easier to do, and probably only a little retrofitting is required after the initial design. But in our cases, we really have to learn from these tests and actually go through this design build test cycle several rounds. And that takes time, and it costs money because it requires resources. And we have to go through this cycle here many times in order to actually reach targets for industrial production and actually meet econometrics that actually can compete with classical synthesis of the chemical that we're interested to produce. But we are advancing on the design tools here, and particularly we're using mathematical models, and I'm not going to talk so much about it here in the first part because I'll come back to it when I'm going to talk about human metabolism. But we're advancing on developing models that can really speed up this design, and we can get better and more precise design tools, and hereby we can then build. Build is interesting. I saw that Emmanuel Charpentier was just elected as a foreign fellow to the Academy because CRISPR-Cas9 technology is an interesting new technology that allows us actually to build much faster than we could before. But there are many other tools that have come along in recent years, so this part here we can do quite efficient. Also here a lot of robotics and different technologies have advanced, so really the bottleneck at the moment is really in this design here, and that's where we are developing mathematical models to advance this. So we are hoping that with these novel technologies that we can really reduce the time from development of this so-called proof of principle strain, and I'm going to give you some examples of where we have developed these proof of principle strains. And then in order to develop that to a level where you have so-called tighter rate and yields, that means that you are producing this chemical sufficient efficient to actually have a commercially viable process. That, as I said, takes many years and many person years also. But with new technologies, we hope we can really carve this down. Several estimates says that this, for some of the existing processes that have been developed, we are talking in the range of $50 to $100 million just to develop the catalyst, which is far too expensive and actually prevents the development of many processes for new chemicals. So we have to reduce this to the order of maybe $5 million for just developing the catalyst. So we have been working on this and particularly looked into yeast, using yeast as a platform organism. And so we have managed to go in and remove ethanol production and produce. We have also worked on improving the classical ethanol production process, but remove also ethanol and produce a whole range of different new chemicals that can find various applications. And some of these can find applications as jet fuels or diesel fuels, can also be blended into gasoline. They can also be used, for example, in cosmetics. They can be used for making acrylates that can go into diapers. Coumaric acid can be used for a number of, it's a scaffold chemical for the chemical industry for making aromatics. Sesquiterpenes, I'll come back and talk a little bit about. Resveratrol also I mentioned, but also active ingredients and recombinant proteins here. And in connection with this, we are developing tools for engineering yeast, but also for designing more efficiently. So why have we zoomed in on yeast? Well, as some people normally say, why not work on yeast? It makes so wonderful products already. But there are a number of reasons to this. Yeast is actually also used as a model organism for studying many fundamental processes in biology. And so many researchers have actually discovered fundamental processes on how human cells are functioning by studying yeast. And if you look back, a number of Nobel Prizes in medicine and physiology have been given based on yeast research. So therefore, we have a very extensive knowledge base on this organism. We have, it's genetically tractable. We can easily engineer it. It is what we call generally regarded as safe because it's already used for so many products. And it's also very robust. It can really tolerate industrial conditions. So it's easy to scale and implement it in industry. And it's already demonstrated and used for a number of different products that are produced commercially. And there's ongoing development for many other novel cell factories. So let me give you just a few examples of what we have been working on with yeast. So in one project that we carried out some years back, and this was in close collaboration with a Swiss company called Firmenich, which is one of the largest companies in the world for making chemicals that are used as ingredient in perfume and fine fragrances. So they are providing these chemicals to the big perfume houses around the world. They are traditionally extracting most of these from plants, but in some cases then you run into limitation because some of these plants that makes a wonderful chemical that has a wonderful smell, you cannot get enough of this plant material. So therefore it's not really scalable. So they were interested to develop a yeast platform to express some of these plant enzymes and then produce, in the start case, some of these chemicals here. And xanthalene is one of the ones we focused on particularly, and that's used in perfumes with sandalwood smell. So what we did was that we did extensive engineering of yeast metabolism. We developed tools for rapid expression of these plant enzymes in yeast, so they were functional. And this led actually to that Firmenich could take the yeast strain that we had developed. They brought it further on for development and now have commercial production of some of these chemicals using yeast-based fermentation. So in the future, when you buy perfumes, you may, some of these ingredients may come from yeast fermentations. Resveratrol is an active ingredient. It's an antioxidant that is in plants, in particular in grape, and where it's sitting in the skin of the grapes. And so therefore there is a higher content of resveratrol in red wine, where when you make the must, you keep the skin in that process. It has a number of interesting properties. It's clinically proven to be beneficial for cancer and type 2 diabetes treatment. but it's not really sold as a drug. There has been some initiative of trying to make drug variants of this, but it's used as dietary supplement and is also in cosmetics. So what we did was that we reconstructed the plant pathway for this. So yeast naturally produce phenylalanine and tyrosine, which are 2 amino acids. And we introduced then enzymes from plants that can convert these compounds into coumaric acid and further on into the final product, resveratrol, shown here. And we developed this process here that was taken on actually by a spin-out company from my research group, Fluxome, that developed a commercial process for selling resveratrol that was later then taken over by the Danish-Swiss company Evolva that is now selling these products, and some of the brands are listed here. More recently, we have worked on a fatty acid production platform, and the objective of that is to be able to produce hydrocarbons using microbial fermentation. And this is challenging, particularly if one do the math on energy content on in sugars compared with oil, because of course sugars have a lot of oxygen in it. And we need to get rid of that oxygen when we have to make hydrocarbons. And because if we don't do that, we cannot make fuels that have sufficiently high energy density to particularly be used in jet fuels. So in order to do that, we can produce long chain of free fatty acids, which are fatty acids. There's still oxygen sitting at the end, but a number of enzymes can be expressed, so we actually take that out and eventually produce the alkanes that can then be used in fuels. One of the later enzymes here turns out to be very hard to express actively functionally in yeast. This enzyme traditionally was existing in various bacteria that has produced all the oil that we're relying on today. But that happened over millions of years. So we are working on improving the efficiency of this enzyme, but right now we have managed to very efficiently produce free fatty acids. Several of the oil companies are interested in that because they can actually do a chemical hydrogenation of the fatty acids, and this is a process that Preem is currently using by taking plant oils from the forest industry in Sweden and making hydrocarbons and using that as blend-in in diesels. So we may end up in having a process where we're producing the free fatty acids and we're chemically converting that into the hydrocarbons. We were interested to see if we could then also make different chain lengths, because the fatty acid that yeast makes naturally have 16 or 18 carbon atoms. But if you're going to kind of cover the whole spectrum, you would also like to have hydrocarbons that are shorter. So we looked into the enzyme, and this is a multifunctional enzyme that makes these fatty acids. So this is like a little nanomachine that is sitting inside the yeast cell, and it actually carries out a number of chemical reaction processes. We went in and inserted a new enzyme activity inside the active site of this enzyme, and hereby allowed that when the carbon chain is growing, and it's growing by 2 carbon every time it clicks around in this enzyme, and when it comes to C8 or C10, 8 or 10 carbon atoms, it's cleaved off from the enzyme. And we were successful to do that to insert this so-called thioesterase activity, and hereby we could begin to produce C8 and C10 fatty acids, and they can then be further converted to alcohols or alkanes. And that's interesting because you go up for much higher energy density than you have in ethanol, for example. And we were collaborating with the French energy company Total on this, but we also licensed this into our spin-out company, Biopetrolia, who's interested to look into also alternative uses of these short-chain fatty acids. We're also interested to go the other way and make even longer chain fatty acids. These are quite rare in plants. There are relatively few plants that makes these long chain fatty acids like C22. But we combined endogenous enzymes from yeast, but also expressed a bacterial fatty acid synthase that actually makes these specifically. and then we're able to make C22, but also convert these further into alcohol, so we get this very long chain fatty alcohol here, which is an ingredient or a component actually of your jojoba oils that is used in cosmetics and is generally limited in amounts. Finally, we have been working on developing also and improving the performance of yeast for production of human insulin. Human insulin is a quite small protein, but we used a technology of microfluidic screening where we, and this was done in collaboration with people at KTH, where we could sort cells, and then we did a genome sequencing of some of the strains that were performing better, and then we could identify causal mutation that actually helps the yeast cells to secrete and send the protein out of the cell. And we are now evaluating this further to see if we can use these yeast cells also to produce other recombinant proteins that are of higher value for the biotech industry. So let me end this part here with coming back to the antibiotics, because what are we going to do about that? I mentioned that there's a major problem about antibiotic resistance. So here comes another benefit from genome sequencing, because traditionally we have been finding microorganisms out in nature and then be screening them for producing new chemicals. But today we can use genomics much more actively, because if we as we did here, we genome sequence a number of different filamentous fungi, and then we found that on average these fungi have more than 50 so-called biosynthetic gene clusters, meaning that they actually have the capacity to produce 50 different chemicals that may have bioactivity. But normally when we screen these fungi, we only find 2 or 3. And that's because most of these are likely going to be latent, and they're only expressed under conditions where we don't really can expose them to. But what we can now do is that we can take out these genes that we identified from this study here, and then we can express them in yeast or in other filamentous fungi, and we can then evaluate these, and this would really advance our chemical space of new compounds that can be screened for antibiotics and other bioactives. So this is just an example of how these clusters look like. They are assemblies of genes that are encoding for different enzyme activities. And what we are currently working on is to carve out and identify which specific activities here, and will we then in the future even be able to, for example, predict what kind of chemistry or chemical compound will be derived from a certain genome sequence. So I hope to end this part of the presentation here that I made it clear that bio-based production can really contribute to establishing a sustainable society, of course in combination with many of the other renewable technologies that we see coming along, photovoltaic, wind, hydro, but biorefineries can really deliver us the chemical building blocks that we are relying on, as well as also some of the liquid transportation fuels that we need, for example, for jet planes. And the latter is kind of interesting because there was a paper published recently in Nature where they were actually studying that not only will biofuels reduce CO2 emission, greenhouse gas emissions, but also it will significantly reduce particle emission and hereby global warming. And this was illustrated here where they were blending flying jet plane with 50% blend-in biofuels compared with standard kerosene, and you could see significantly reduced particle emission from the blended-in diesel or jet fuel. So now let me move on to see if we can use our knowledge on yeast metabolism to study human diseases. And this is approximately where I was when I came to Chalmers in 2008. I'm a chemical engineer of training, and I did most of my career at the Technical University of Denmark. It was relatively hard in a small country like Denmark. Sweden is a little bit bigger, at least landmass. It's much, much bigger. But it's hard in a small country to grow out of kind of how you are categorized as a researcher. And so, therefore, it was very hard for me to actually go and start to do work on human metabolism. But when I came to Chalmers, nobody really knew what I had been doing before, so they were very open-minded to start collaboration. And so we started a number of research collaborations of beginning to use the same modeling concept for studying human metabolism. And we have studied a number of different human metabolic diseases, but I'm only going to talk about some results from cancer here. And this is particularly interesting in this move forward towards what is referred to as personalized medicine, because traditionally the pharma industry has been relying on what you call blockbuster drugs, where you are having one drug fits all to treat disease. But in the future, we can use, again, technologies from the genomics revolution to stratify in much more detail people into groups and then treat individuals in these groups with different types of medicines. But in order to do this efficiently, after we've done maybe a very thorough stratification, because this would be too expensive to scale up, we need to identify biomarkers. So really biomarkers is at the core of advancing personalized medicine forward. And so let me talk a little bit about the modeling technology that we have developed to study this. So what we are doing is that we are working on these so-called genome-scale metabolic models that are comprehensive reaction libraries of the cell that we are looking at. So we're looking into all the different biochemical conversion processes, all the enzymes that catalyze these reactions. These enzymes are, of course, links to gene in the genome. So now this gives us a link between the genome and the enzymes and the chemistry that is actually carried out in this cell. That gives a so-called metabolic network. I showed you earlier how that looks for yeast, but it also gives a mathematical representation that we can use for simulation. So what we did was that we comprised and collected everything we know about human metabolism into a so-called generic model. So this is a model of all the reactions that can happen in many of our different cell types. And then we developed also an algorithm where we can take information about which genes or proteins are expressed in a specific tissue or cell type. And now we can then make tissue-specific models, for example, a model for liver or one for muscle or adipocytes or whatever. And we can also do this, of course, for differentiation, what is happening in differentiation when, for example, a cell is going from healthy state to a cancer cell. So we work very closely with Matthias Ullén here at KTH on this. This is just an illustration where we did a heat map of the activity, and here's shown for 32 different tissues, and here's shown groups of reactions. What you have first here is presence with red. This is liver. Liver have almost everything of metabolism. It's extremely diverse in its metabolism. Whereas if we go to other tissues, you can see they have reduced, and particularly this part of metabolism up here, they don't have at all. So, but how can we use this then to identify biomarkers? So what we have is that we have this metabolic network. We can now take, for example, transcription data. That means which genes are turned on in a cancer. We can overlay that here and then particularly compare with whether there are some parts of the metabolism that are now turned on in the cancer that is not active in the normal cell. And then we can begin to look for whether some of these metabolites that are associated with that part of metabolism are actually produced and can maybe be found in blood or urine. And that could then hopefully lead then to identification of the biomarker. We started this work actually because we were interested in a more fundamental question, whether cancer metabolism is more similar among cancers compared with actually with the tissue of origin. And so the PhD student who started to do this work started to look into 10 different cancers. But what he found was that there was one cancer that was sticking out and completely separate from the others, and that was clear cell renal carcinoma, the most predominant form of kidney cancer. And that was separating here in this so-called PCA analysis here completely from all the other cancers. It turned out that there are a number of reasons for this separation here, but what we found in connection with this also was this so-called subnetwork, part of the metabolism that is significantly altered in this type of cancer here, and that is involved in heparan sulfate and chondroitin sulfate biosynthesis. And so here you again see a network here. It's very hard to dissect, but what we can do is that we can overlay it on these more traditionally biosynthetic roadmaps that you have in this in textbooks, where you can see here you have the different chemicals being interconverted into each other and so on, and you enter some down products here. You have the different enzymes here. And when we have red, that means that there's more of this enzyme. When you have blue, you have less of this enzyme. So you can almost imagine that if we look for some of these metabolites over here, we may expect that there is a higher level of these in the tumor, or maybe even the associated urine and blood. And there's maybe less of these over here. We confirmed these experiments with actually looking for the enzymes, and that was okay. And then we started to measure these associated metabolites. And we identified that we should analyze 20 of these metabolites. That was kind of interesting because it was a little bit difficult. I said it normally goes very easy for us to get access and work with clinical researchers here. But in this case, it was a little bit hard to convince actually to give plasma and urine samples. But the PhD student, Italian, was very entrepreneurial and identified via his uncle's former cancer clinic in Italy that we could get samples shipped to Sweden for analysis. We were then looking for someone to help us with analyzing these chemicals here. That turned out to be a little bit difficult again. So we ended up finding a lab in Italy where we sent down the samples to Italy and for analysis. And then we got analysis of these different chemicals here. Here you can see that, for example, in the tumor, this one is higher, and there's error bars here because we are measuring in several different patients. For some of them, there's no really difference and so on. But what we can now do is we can take a computer algorithm to try to search for differences that there is between the healthy and the tumor tissue. And then we can calculate a so-called score. And here you can see the score here. This is for metastatic clear cell renal carcinoma here. Here you have healthy. You have clearly above the score here 1. This is based on combined. This is based on only plasma, and this is based on only urine. We can do an evaluation of this score by a so-called ROC curve, where we look at the area under the curve here. If that area under the curve is 1, that means we have a perfect prediction, and we actually have that in the combined for this initial clinical study. And this we validated in a follow-up cohort, and we have actually now validated this for clear cell renal carcinoma in more than 500 subjects, and the result still stands like this. And so there's a lot of interest in the clinical sector for implementation of this for different applications, because it can it warrants timely switch of drug line. So a lot of the cancers do not respond to the first line. You can now monitor much more frequently than you can with normal line of treatment. And so you can shift to another drug line. You can also spare unnecessary surgeries. 20% of renal masses are benign. You can save these surgeries. You can also monitor treatment of recurrence. Because 20% of confined cancers return as metastatic. And it can also hopefully, and this is something that is harder, but it can hopefully lead to earlier diagnosis. And this is particularly a problem in clear cell renal carcinoma that is detected often very, very late. So we got excited about this, so we started to look at whether was this something that was unique for clear cell renal carcinoma, and so we actually looked into other cancers also, and found that, and this was prostate cancer, that we found a similar observation that we can again separate. It's a different score, but it's the same 20 metabolite we're analyzing. And what you can see here is that when we take the score for the prostate cancer, and take the measurements from the clear cell renal carcinoma, it doesn't come out. So it's a different profile of these 20 different metabolites that we see in these two cancers. So we can even distinguish between two types of cancers in this case here. So let me end. We'll talk about another exciting applications of our models, and that is to study the complex system that is occurring in the human gut microbiome. So we all have about 2 kilos of bacteria in our intestinal system, and these bacteria are playing a very important role in helping us in digestion of food, as well as also providing vitamins and other important nutrients for us. There's been some studies about that we contain 10 times more bacteria than human cells. There's a big change in this also from early stage in life throughout, where we are having changes in that, and that may also be associated with some of the risk developments in elderly people. There's been a lot of reports about link to disease, but they are, as stated here, they are links to disease, associations. There are correlative studies. There are no mechanisms. We really need causal information in order to move towards intervention for disease treatment. So we've been involved, and this has all been in close collaboration with Frederick Beckett at Sahlgrenska in Gothenburg in studying the human microbiome using metagenomic sequencing. So we developed all the bioinformatics pipeline for taking the data that comes out from genome sequencing and taking that into biological interpretation. And these are very large data sets that we are getting. So we can see here the number of data points that we are getting from this kind of study. So one study we did was that we looked into abundance of different species. So here you have different bacteria that could be identified. And this was looking into cohorts that was either type 2 diabetics or healthy. And what you could see was that you have a number of organisms here that negatively correlates. Blue means negatively correlates with a number of these different blood markers here. You have some red ones here, but these are the good ones that you would like to have, HDLs, for example. And then you have here what is bad profile. All these bacteria correlate positively with these blood markers. So I guess I said this is kind of indicative. But what is of course interesting is could we identify some of these bacteria here and get them to colonize into people with type 2 diabetes and hereby change or maybe revert. Maybe we cannot cure the disease, but maybe we can revert when they go to a stage of impaired glucose tolerance. So we looked into using modeling for this, and in this case here we were showing that we could actually model the change that is happening in amino acid production by these bacteria when you undergo a change in diet. And so this is a complicated study here, but we developed mathematical models for 10 different bacteria that lives in the human gut and how they interact. We then integrated also information about the nutrients that they get, the patients are getting, or the persons involved in this study here. And we could show here that there is this change in the amino acid levels, and actually people respond differently depending on how their gut composition was at the outset of this study here. So this was a very important pilot proof of that we could actually begin to predict what are the changes that are happening in response or depending on the gut microbiota. And so we're now looking into if we can classify these as core, because if we are beginning to use this actively going forward for treatment of disease, we will need to make sure that the bugs that we are giving are actually colonizing stably. So maybe we should not just give only one, but we should give several species that should interact in the right way. So we started to look into what are really core, generalist and specialist in the gut microbiota. And then we can begin to find core bacteria that are always correlating with a set of other bacteria. And if we give these, they are likely to correlate and actually colonize and recruit others that may then actually change the microbiota. So this is still early time on this, but I'm quite sure that we can actually use this, and we did some initial study with the Bill and Melinda Gates Foundation of actually looking into the gut microbiota of stunted Malawian and Bangladeshi cohort that were not responding to ready-to-use therapeutic foods. So they were still not actually having sufficient energy uptake. But could we in the future begin to use probiotics, new generation of probiotics, to treat diseases like this? So I hope I convinced you all that studying metabolism, at least has been very good for my career. I'm standing on the stage tomorrow. But also understanding metabolism, it really allows us to address many of the grand challenges that we are facing in the future. So with that, thanks. I'm not going to mention all the fantastic students and postdocs and collaborators I've had throughout my career. I often say that that is one of the best part of my job. That is that I am meeting young, smart people constantly, and when they start to be, when you are in industry, after a couple of years they are looking for new challenges and you have to find them in the company, then they all normally leave my group because they move on, and they're supposed to leave on, and then new young smart people come, and that's fantastic to see them grow in that process here. And of course the various funders, and thank you for your attention.
Thank you. Truly impressive piece of work. I mean, I'm really, really impressed by how you link the fundamental understanding with... unfortunately, we don't have time since many of us have a dinner to attend also. But I will ask you one question with the privilege of chairing this event. I guess that when you correlate with like blood biomarker that you know are correlated with the pathogenicity of a disease, you cannot know from the beginning whether they are the cause or an effect of the disease. I mean, they correlate, but of course, by taking the microbe, you will find out. But what's your comment to that? When you correlate, you don't know whether it's the cause.
Yeah, so for biomarker identification, it doesn't really matter so much as long as it's consistently, and validated in new cohorts and so on. That is generally applicable. For treatment of disease, and so this is one thing that we are puzzled about. For example, on the glycosaminoglycans, heparin sulfate, and so on. Are they something associated with the disease, or is it something that is happening because the tumor needs to expand in the surrounding and the connective tissue and so on, and therefore you get this remodeling of this, and that could be also why it is actually different for different cancers, because that remodeling may be different depending on the tissue of origin. But so therefore, that's probably not going to lead to any disease treatment. But that's good enough if we can use it for classification and diagnosis. For the gut bacteria, we of course need to know how and what is the culprit of this. And I can say that we have some studies on metabolomics in collaboration again with Frederick Beckhead, where we are finding gut metabolite, bacterial metabolite, that are actually impacting and stimulating receptors in the liver, for example, and hereby may impact the development of type 2 diabetes.
Yeah, I'm sure that your extremely impressive research, I'm very proud that you are awarded the gold medal tomorrow, but also I'm sure oncologists really are excited about this, because it gives some new opportunities to study these cancer diseases. So I think we should give Jens our big hand and thank all of you.